Okay, can somebody confirm you can hear me?
I can hear you.
Thank you. I think I've only heard two people join. Let's check on that. I can't get to the screen now. Yeah, there's only two people who joined. I don't know why there's so few today. Let's continue. Today is February 20th. Are there any questions before I begin? All right, we're supposed to be working on Chapter 8, Microbial Genetics. We talked last time about RNA transcription, and hopefully I got that link working now. Did I show you the RNA transcription last time?
Yeah, we left off at the translation.
Yeah, okay, great. Let me get rid of that. That's not true anymore. I put new links in there. Oh, actually we have it, but we have here. Wasn't in the right place either. All right, translation is our next topic. Translation is the synthesis of protein by ribosomes using the information in messenger RNA, which that information, of course, comes from DNA. Messenger RNA is translated in codons. A codon is a group of three nucleotides. So you just start with one nucleotide, add two more, and then you read it as a codon. And three nucleotides, or a codon, makes one amino acid. So translation of messenger RNA does not begin until the ribosome reaches the start codon. The start codon is always AUG. AUG does serve a double function, and that is it tells the ribosome start protein translation here, and AUG also encodes for the amino acid methionine. And then the ribosome starts reading the messenger RNA in groups of three called the codon, filling in the amino acids until the ribosome reaches one of the three stop codons, and that will be a group of three nucleotides that tell the ribosome stop protein translation here. And there's three of them. You don't need to know the stop codons. You should know the AUG codon because it's the only start codon. But once the ribosome reaches either UAA, UAG, or UGA, you don't need to know these. It will stop protein translation. So here is what the codons code for. Let me see if I can blow this up. And you'll note that there is more than one codon for most of the amino acids. Like phenylalanine has two codons, UUU and UUC, which code for the same amino acid, phenylalanine. Leucine, on the other hand, has six codons that code for that one amino acid. Valine has four different codons that code for that one amino acid. There are only two amino acids that have a single codon coding for that amino acid. One of them I've already mentioned to you, and that is the start codon coding for methionine. I'm not seeing it up here now. It's AUG coding for methionine. That one I have memorized, but I'm not finding it now. The other one is tryptophan. You don't need to know that. There's only one amino acid for that amino. There's only one codon for that amino acid. So most of the codons, all but two of them, and I can't find methionine. Anyone see methionine? Funny. Oh, there it is, right here. AUG codes for methionine. It also codes for starting protein translation at this amino acid. Those are the only two that have one codon coding for that amino acid. Most of the others have 2, 4, or 6 codons coding for that amino acid. So there are 20 different amino acids in humans, but there are 64 possible codons, meaning some of the codons code for more than one amino acid. And this is known as degeneracy of the code, where more than one codon can code for the same amino acid. Let's talk about protein translation. I think I'll start with the movie first. Protein translation begins when the ribosome assembles on the messenger RNA, and then tRNA brings the first amino acid in. All three RNAs take part in protein translation.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon.
Okay, you do not need to know the P site and the A site. You just need to know there are 2 sites in the ribosome that tRNA can come through the ribosome and then hydrogen bond to the messenger RNA. And that's why the AUG has a anticodon on the tRNA, which is UAC right there.
The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters.
And then let me briefly go over something right here.
RNA codon.
There we go. What's removing the methionine from the first tRNA and then linking the methionine to the growing polypeptide chain here is actually an enzyme that is an RNA enzyme. It's part of the ribosome. So it's RNA in the ribosome. And that's the only
The methionine is then
that's the only RNA enzyme we'll talk about in this class.
transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
All right, any question about that little video? I think I'll keep this open because we've got other videos, I think, in this lesson. Let me close some of these things down. Oh, that's not open, sorry. All right, let's look at translation in a little more detail. Translation is the flow of information from messenger RNA to the protein. Of course, the information originally came from DNA. Translation involves the messenger RNA carrying the code to make a protein, the two ribosomal units, and then tRNA, which brings in the correct amino acid. And the first amino acid on every protein is, of course, methionine, because methionine binds to the tRNA that has the anticodon UAC, which this UAC anticodon then can hydrogen bind to the start codon AUG. And the ribosome, we say the ribosome reads the messenger RNA, but in reality, the ribosome is putting everything together, lining up the sites on the ribosome for the AUG for the tRNA to then come in and bind to the codon. So the correct amino acid will be added because the correct amino acid will have the correct tRNA with the correct anticodon, which will then bind to the hydrogen bind to the codon. Any question about any of that? All right, so the ribosome forms on the messenger RNA, and it has two sites that allow tRNA to come in and then hydrogen bind to the messenger RNA. And the sites in the ribosome sort of stabilize the tRNA because, as you know, the three hydrogen bonds between A, U, and G, and UAC, meaning A binding to U, A binding to U, G binding to C, it's not going to be a very strong bond. And so the ribosome is putting it all together and stabilizing it. And then the second tRNA can come in as the correct anticodon, which will bring in the correct amino acid to the second codon. Then the enzyme of the ribosome, which is an RNA enzyme, removes the first amino acid from the first tRNA and then links that amino acid to the amino acid on the second second tRNA. And now that will release this tRNA. So it'll come out. And when that happens, this ribosome will then move down one position. And so this tRNA with the two amino acids now moves from this site to that site. And that will allow the next tRNA to come into the second site of the ribosome, where it has there. And then once again, this amino acid will be removed from that tRNA, and then this growing polypeptide will be added to that newly added amino acid. That tRNA will then come out, which will allow the ribosome to move down to the next codon, allowing the next tRNA to come in. And that process continues as the ribosome moves down the messenger RNA one codon at a time until the ribosome reaches a stop codon. When that happens, the protein or the growing polypeptide chain, whatever you want to call it, is released from the tRNA. The ribosome dissociates and falls off the messenger RNA, and that will release the last tRNA, stopping protein translation. So the stop codon does essentially all of those. It codes for the ribosome to fall off the messenger RNA, codes for the polypeptide to be released, and then the last tRNA to fall off the codon, which is shown here. That growing polypeptide chain, once the last amino acid is added, we now call it a protein. Before that, you don't call it a protein. You can call it a growing protein. It's best to call it a growing polypeptide chain. All right, any questions about protein translation? I want to give you a chance to ask questions because I'm going to now talk about some complications of protein translation. Here you can see protein translation actually occurring in a cell. I think this is a bacteria cell, but I don't know for a fact about that. Yeah, this isn't a bacteria cell because you won't have one of these complications happening in a eukaryote cell. Anyways, this is an electron microscopic image of protein translation. And there are several things you can note here, and that is in the same gene, we are transcribing more than one messenger RNA at the same time. And that's why this is the longest messenger RNA. And these over here are much shorter. And as you go up, they get longer. That one's hard to see, but that one is longer than that one, which is, it's hard to see, but that's actually longer than that one, which is clearly longer than that one, longer than that one, then longer than that small one. And that's because the RNA polymerase started over here. And as it works this way, it makes a longer messenger RNA. And this is the artist's rendition right here. Let me blow this up. So the blue line is the messenger RNA. And as you move this way, you get a longer messenger RNA. then right here, which would be where the gene starts, about right there on here. So that's one complication that you have in eukaryotes and in prokaryote cells, and that is one gene can have more than one messenger RNA being made at the same time. And all you do is you have 1, 2, 3, 4 here, 1, 2, 3, 4, 5, 6, 7, 8 different RNA polymerases bound to the same gene, and they start here and they move this way to make the longer messenger RNA. Any questions about that complication? In prokaryote cells, we have another complication occurring, and that's actually shown by these globs on the messenger RNA, or these red globs here. And that is, you can have protein translation begin in a bacteria before RNA transcription finishes. So this very short, or that very short there, messenger RNA has these globs on it. These are ribosomes, and they are starting to make the protein for this messenger RNA. And the messenger RNA has not even been finished being transcribed yet. You have this complication happening in prokaryotes because this is happening in the cytoplasm, and the ribosomes are in the cytoplasm, so is the DNA, so is the messenger RNA. So they're all together. And so protein translation can begin before RNA transcription finishes. Any question about any of that? And then the other complication is you can have more than one ribosome bound to the same messenger RNA. So here we have 2 ribosomes bound, probably 2 there, maybe 3 or 4 there, 3 there, 4 there, 5 there. And each of them is making a protein. So that's the other complication you have in a prokaryote cell. We do not have this last complication in a eukaryotic cell because the transcription occurs in the nucleus of a eukaryotic cell and protein translation occurs in the cytoplasm. Any question about any of that? But in bacteria, you can have protein translation occurring at the same time RNA transcription is occurring, and protein translation begins before transcription ends, and you can have more than one protein being made from the same messenger RNA. In fact, you can have multiple, like right there. It's got, I don't know, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, about 20 different ribosomes bound to this one messenger RNA. Any question about any of that? All right. So I mentioned that eukaryotes are a little easier, and that is they don't have the complication of protein translation occurring before transcription finishes, because transcription happens in the nucleus, and then the messenger RNA moves into the cytoplasm out of the nucleus, and in the cytoplasm is where protein translation begins. However, in eukaryotes, we have another complication. And that complication is that eukaryote genes have both exons and intron. An exon is a region of a gene that actually codes for the protein. The intron is a region of the gene which does not code for the protein. And so when the messenger RNA is first made from a eukaryote gene, it will be transcribed into regions of exons and regions of introns. These introns are spliced out of the gene or the messenger RNA. Spliced just means cut. And then the exons are spliced together, so they're reassembled. And this initial RNA transcript cannot leave the pores of the nuclear membrane. So there's a safety checker on the pore that will not allow a messenger RNA to leave if there are introns in that messenger RNA. It's not until the introns have been spliced out and then the exons spliced together that the final messenger RNA can leave the pores of the nuclear membrane and then go into the cytoplasm to be translated. And that's the complication we only have in eukaryotes. Any question about that? In bacteria, we don't have introns inside the gene. I think archaea do, but in bacteria, you do not have introns in the gene. But in eukaryotes, we do. And just think of the intron as another region of non-coding DNA. Any question about any of that? Remember, the eukaryote DNA is mostly non-coding DNA. There are regions inside a gene which do not code for the gene product, meaning the protein, but there are large regions in between the genes which do not code for protein or whatever the gene's coding for. And that's not to say that region does not code for anything. At least some of the non-coding DNA codes for the DNA to wrap on itself when the unwound DNA is dissolved in the cytoplasm. Some of the non-coding DNA actually codes for the DNA to come together and form the chromosome. So that's not to say that the uncoding DNA doesn't code for anything. It can have some function. And then obviously it has the function of holding the DNA together. Any question about RNA processing in eukaryotes or anything else? If not, let's show a little chapter summary. We've talked now about the structure and function of the genetic material, talked about the flow of genetic information. We've talked about DNA replication, RNA transcription, and protein translation. So we're going to change gears now and talk about the regulation of bacterial gene expression. On the side, we'll talk about eukaryote gene expression. We won't go into it in any great detail. To talk about bacterial gene expression, we'll talk about operons and what the operon model is of gene expression. We'll then change gears again and talk about mutations. And then lastly, we'll talk, not lastly, but second to the last, talk about genetic transfer and recombination. And then lastly, we'll talk about genes and evolution. A very slight topic. It's only one slide. Any question about what we're going to do? All right, I'm not hearing anything. Any questions? Let's talk about the regulation of bacterial gene expression. One way to regulate the contents of a cell is to control gene expression. If you control gene expression, you can control what products are made in a cell. Some genes are always turned on and are expressed at a fixed rate. These are called constitutive genes also called housekeeping genes, or constitutive enzymes. An enzyme which is always made by the cell at a fixed rate. Other enzymes are only expressed when they are needed. So when the cell needs it, the gene is turned on. When the cell doesn't need the product, it turns the gene off. You can control the genes by two ways. You can have a repressible gene, and you can have an inducible gene. And there are repressible enzymes. These are enzymes which are normally turned on, meaning the gene's normally turned on, and the enzymes are made. But you can turn the gene off, so you stop making the enzyme. And then inducible enzymes are enzymes which are normally turned off, but when the enzyme is needed, the gene can be turned on. So a repressible and inducible gene is regulating gene expression by a pre-transcriptional control. We're only going to talk about inducible operons, which means we're going to talk about inducible genes. We're not going to be talking about a repressible operon. It is similar to an inducible operon. We're not going to talk about it. You will not be tested on a repressible operon. Any questions about that? There are other pre-transcriptional control mechanisms, such as epigenetic control. And epigenetics is a topic that has been studied now for at least 50 years, maybe it's as long as 100 years. I'm not sure when that's been first studied. But it's a very hairy subject that geneticists do not have a good understanding of even today, where the gene is controlled by some epigenetic means, meaning other than an easy genetic control mechanism. I don't know how to word it other than that. So it's a good thing we're not going to talk about it because geneticists don't really understand how epigenetic means control gene expression, at least in the step-by-step way. And then there are other ways to control gene expression. You can have post-transcriptional control, where the gene is transcribed, and then you make the gene product like an enzyme, and then you control that enzyme after it's made. Like one thing you can do is inactivate the enzyme. That is controlling gene expression after transcription, post-transcriptional control. Another way you can do that is by degrading the enzyme. That also controls gene expression post-transcriptionally. And we're not going to talk about that. You're not going to be tested on that either. So the only one we're going to talk about are inducible operons, meaning inducible gene expression. Any question about any of that? All right. So to talk about gene expression, we need to talk about operons. We're mainly going to talk about bacterial operons. You can think of an operon as a super gene. An operon does have the gene of interest. They're called structural genes. A structural gene is usually what we mean when we talk about a gene. So, for example, when we're talking about the lactase enzyme, it is made by the lactase gene, which is actually the Z gene, which is a structural gene. Come on, mouse. It's a structural gene in the operon, coding for the enzyme lactase. Any question about any of that? So normally when we're talking about a gene, we're talking about the structural gene. However, around that gene, we have other genetic material like in the lac operon, we actually have 3 structural genes together in this one operon. And then we have controlling regions around the structural gene that control the gene expression of this structural gene. One that's not shown here is at the very end of the operon. It is the terminator, stopping the expression of the gene, meaning stopping protein transcription. And then another region of the operon, we've already talked about, it's the promoter. This is the region on the DNA that RNA polymerase binds to the DNA. And then the RNA polymerase moves down the DNA to transcribe the structural gene. And for now, I'm just going to talk about transcribing structural gene Z. Another controlling region is the O, or operator. And this is the region of the operon which allows the turning off or the turning on for this operon. So the operator has a protein bind to the operator, and if the protein binds to it, you turn off gene expression of the whole lac operon, or specifically of the Z lactase enzyme. And then when the protein falls off the operator, the gene can be transcribed, and we say that the gene is turned on. Any questions about any of that? Anyways, that's the operon, having a promoter, an operator, one or more structural genes in a bacterial cell, and then the terminator. Come on, mouse. Terminator right here. Those are the operon of a bacterial gene. The operon of a eukaryote gene is very similar, with one major difference, and that is eukaryotes have only one structural gene for each operon. Any question about any of that? All right, so how does regulation of the lac operon occur? It happens by induction, but it could happen by repression. We're not going to talk about repression. Induction is where the gene is normally turned off, but when the gene is needed, it will be turned on. So that's induction. Repression is the reverse. Repression, the gene is normally turned on, but when the gene is not needed, it is repressed and turned off. The lac operon is the only operon we're going to talk about. It is in a bacteria responsible for metabolizing the sugar lactose, including making the lactase enzyme. And it is an inducible operon. So normally, the I gene shown here makes a repressor protein shown here, this M. It's the active repressor protein. And when it's active, this repressor protein will bind to the operator. And when that happens, when the RNA polymerase binds to the promoter and then moves down the DNA, it'll get stopped by the repressor protein. And therefore, the Z or lactase gene cannot be transcribed, and the gene expression is turned off. Any question about any of that? So that's how induction works. And in E. coli, let's say it's E. coli and it's on an apple. The E. coli on an apple is not going to see the sugar lactose. So the E. coli will turn off the expression of the gene because it doesn't need the lactase enzyme or any other of these structural genes in the lac operon, because the sugar lactose is not going to be on an apple. Right? Any question about that? And if you think about it, E. coli, any place it's going to be, it's not normally going to be around the sugar lactose. Because where is the sugar lactose found? Come on, somebody's got to know that. Where do we find the sugar lactose? Well, think about it, because I'm not going to tell you. E. coli is not likely to see the sugar lactose unless it is in a dairy farm or in somebody's gut that is drinking dairy products or eating dairy products, because the sugar lactose is not commonly found by E. coli. So E. coli turns off the expression of this gene by induction. Any questions about any of that? And you guys are going to have to answer where the sugar lactose is found. And how it turns it off is this other gene over here, the I gene. Let me back up a minute here. So we're saying this I gene right next to the lac operon. Let's go there. It's a little better. So here is the operon. This I gene is not part of the operon, the lac operon. It actually is its own operon, and it makes the regulatory protein, which regulates the expression of this operon by binding to the operator. Any questions about any of that? Now, normally, the regulatory gene is found nowhere near the gene it regulates. In fact, in eukaryotes, the regulatory gene is actually found most of the time on another chromosome than the gene it regulates. In bacteria that largely have one chromosome, the regulatory gene is not normally found close to the gene it regulates. In the case of the lac operon, the regulatory gene is right next door to the gene it regulates. But that's not usually the case. All right, so that explains why the lac operon is turned off. And when it's turned off, the Z gene, the Y gene, and the A gene are not transcribed. The Z gene, I've already told you, encodes the sugar lactase. The Y gene is permease, which is the transport protein that brings the sugar lactose into the cell across the cell membrane. So the Y gene codes for the transport protein to bring the sugar into the cell. And you don't need that gene when there's no lactose outside the cell. And then the A gene is involved in the metabolism of lactose. And when you break down the sugar lactose by the enzyme lactase, which the Z gene does, you make glucose, which the cell can use, and then it makes beta-galactose. Beta-galactose needs to be converted into glucose before it can be metabolized. And the A gene helps convert beta-galactose into glucose. So all three genes are involved in the metabolism of the sugar lactose. Any question about any of that? All right, so we need to have some way of turning on the lac operon whenever the cell sees the sugar lactose. How we turn on the expression of the lac operon in the presence of lactose is we have an inducer which can bind to the active protein made by the gene I, the regulatory gene. And the inducer binds to the active protein, inactivating the repressor protein. When it is inactivated, the repressor protein cannot bind to the operator. It turns out the inducer is allolactose, which is the isomer, meaning the left hand of the right hand or the right hand of the left hand. Allolactose is the isomer of the sugar lactose. So whenever you have the sugar lactose around, you will have allolactose around because it's the isomer. And if you don't know, an isomer can switch from one form to another. And so even if you were to purify and put in only the sugar lactose, some of that lactose would eventually convert to allolactose. And allolactose will bind to the active protein made by the regulatory gene, inactivating the protein, so this protein cannot bind to the operator. If there's no repressor protein binding to the operator, then when the polymerase binds to the promoter, it will move down the operator and then transcribe the lac operon, transcribing the Z gene, the Y gene, and the A gene. Any questions about any of that? Let's watch the little video. I tried to open that earlier, and I couldn't get it open.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from.
That is a little bit of an oversimplification. The repressor molecule is not released by the operon when the repressor protein binds to the inducer, which is allolactose. It's a little more complicated than that, and I'll discuss that after the video.
From the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein.
Let me mention that beta-galactosidase is another name for the enzyme lactase. So those are the same enzyme.
Into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
All right, any questions about that video? Let's talk about that in a little greater detail. Let me blow this up again. So what do you notice about something unusual about the transcription for the lac operon? Well, how many genes do we get out of the messenger RNA?
3.
Yeah, 3. We've never seen that before, but in the case of bacterial operons, you will transcribe a messenger RNA that will code for 3 different proteins, at least in the case of the lac operon. And that will happen if ever you have a operon that has more than one gene, structural gene in the operon. In eukaryotes, you never have that because, like I stated, in a eukaryote operon, you only have one structural gene in the operon. Now, obviously, humans can digest milk, which has the sugar lactose, right? And we do have the gene for lactase. We have the gene for permease, which brings the sugar lactose into the cell membrane. And then we have the A gene, which converts or helps converts the beta-galactose into the sugar glucose. So we have all 3 structural genes. It's just that they are not on the same operon. They each have their own operon. So in humans, we have 3 different operons to digest the sugar and lactose. And in E. coli and bacteria, they only have one operon because the related structural genes are put together in bacterial operons. So that's one difference there, and that's the unusual thing. The other unusual thing is I mentioned that the inducer, allolactose, will bind to the active protein, making it inactive so it can no longer bind to the operator. And that's fine if the operator doesn't have anything on it, but if it's bound to the active protein is bound to the operator, how does the active protein come off the operator so that the lac operon can be turned on? Anyone have a guess? It's not that the repressor protein binds to it, because when the inducer binds to it, because when the repressor protein is bound to the operator, the inducer cannot bind to it. What happens is the binding of this repressor protein to the operator is by a stoichiomic reaction, and every once in a while, the repressor protein will fall off. And if there is the inducer around, allolactose, it will then bind to the repressor protein when it falls off. And now there's no active protein to bind to the operator. And then the lac operon will be turned on. Here's another question for you. Let's back up. And that is what happens to this RNA polymerase when it gets stuck at the operator because it can't proceed further because the repressor protein is bound here. Is this RNA polymerase stuck here indefinitely until the active protein falls off? Anyone have a guess? It's either yes or no.
Yes.
No, it's actually not stuck there forever. The polymerase also binds to the DNA by a stoichiomic reaction. So every once in a while, the RNA polymerase will fall off the DNA, and then the RNA polymerase can bind to another promoter and then transcribe that gene. It could also bind once again to this promoter, but it'll stop at the operator if the repressor protein is bound there. So both of these reactions are stoichiomic reactions, and every once in a while, the protein will fall off. And that makes sense because you don't want the RNA polymerase to be stuck here, because it could be stuck here for the lifetime of the E. coli. And you don't want that happening. You want that RNA polymerase to be able to transcribe the DNA. Any question about any of that? Anything else I need to talk about that? I think that's it. All right. Any questions about, specifically about the lac operon or about operons in general? The nice thing about the lac operon is whenever the sugar lactose is not around, the repressor protein will bind to the operator, turning off the expression of the structural genes in the lac operon. So we do not make lactase if there's no sugar lactose around. And the cell doesn't need the enzyme lactase around if there's no sugar lactose around. But if sugar lactose is around, we'll have allolactose around. It'll bind to the repressor protein, which will turn on the expression of the gene because eventually the repressor protein bound to the operator will fall off. Allolactose will bind to it, and then the polymerase can transcribe the Z, the Y, and the A gene. Any question about the lac operon? All right, if not, let's move on to mutation. A mutation is a change in the genetic material. Mutations may be one of three things. They can be a neutral mutation, neither benefiting or hurting the individual containing the mutation. They can be a beneficial mutation, or they can be a harmful mutation. I'm sure many of you can tell me a harmful mutation. Can anyone mention a harmful mutation?
Is it cancer?
Yeah, that's several mutations, but yes, cancer would be a bad mutation. Let's give a more genetic mutation, meaning a disease that's caused by a genetic mutation.
Sickle
Can
cell.
anyone mention one of those?
Sickle cell anemia.
Yeah, sickle cell anemia is a good one. And that is, we have a mutation happening in the DNA, or you inherit it from your parents, and it causes sickle cell anemia, which is a harmful mutation to anyone who has two genes with this mutation. A beneficial mutation, I only know one off the top of my head, and that is there was an individual who was keeping fruit flies in his lab, and he was kind of lazy, and so he wouldn't change out the bottles of fruit flies. And fruit flies, when they larva gets ready to transform into the fruit fly, it'll become a pupa, and it has to climb to the side of the jar, and then it's in the pupa stage, and the pupa stage will go through metamorphosis, and then it'll change into the adult fruit fly. And anyways, the guy didn't change out the bottles of fruit flies, and so on the entire side of the bottle there would be all these pupa, and eventually there would be so many that there wasn't any room for future fruit flies to crawl up and then form a pupa. And this individual just continued this way, and he had a mutation happen in his population of fruit flies. And the mutation was, instead of the larva crawling to the side of the glass and then forming a pupa, some of the larvae actually crawled up to the cotton. Let me see if I got this is close enough. It might be cotton, I don't know. He had a cotton swab, I don't know if you can see this, inside the bottle, and they would actually climb up to the cotton swab and then form the pupa in the cotton swab. And there were so many pupa on the side that the few that had that mutation increased in population in his fruit fly lab. And then eventually most of the population became the ones carrying that gene, because later on, the only ones that were reproducing were the ones that could climb up to the cotton swab and then have their pupa on the cotton swab. So that was a beneficial mutation in his population of fruit flies. Any question about any of that? Actually, the reason why sickle cell anemia occurs is because in the homozygote, it is a detrimental, meaning it's harmful for the individual. But in the heterozygote, one individual has only one gene, and the other gene is the normal gene for hemoglobin. These individuals benefit, and that's because those individuals are much better at clearing malaria from their body than a normal individual who has normal hemoglobin, meaning two genes for normal hemoglobin. So in the environment where there's malaria around, people who have one copy of the sickle cell anemia benefit. They're more likely to survive malaria. They're more likely to reproduce. The trouble is, the individuals with two sickle cell anemia genes that's a harmful mutation. You got two of them, and anyways, that's just a complication of sickle cell anemia. When you have two of them, it's harmful. When you actually only have one, and you're in an environment with malaria, it is actually a benefit to you, because you're more likely to throw off malaria. And you don't need to know why, but it's because individuals who are heterozygote, the red blood cell, which becomes infected with malaria parasite, will sickle, and then the red blood cell will be removed from the body, and that will cure malaria from the patient. Any question about mutation so far? All right. A mutagen is an agent that causes mutations. Can anyone name a mutagen? Anyone? Aren't some of you smokers? Smoking is a
Nicotine?
mutagen, which actually is one of the more powerful mutagens that humans get exposed to. You can also get a mutagen by going into sunlight and burning your skin, getting UV rays. That's a mutagen. Other chemicals can be mutagens.
Like diesel, could that be a mutagen?
Diesel itself, I don't think is a mutagen, but there are products in the diesel, which
Okay.
I think are the mutagens. And um, it might even be possible that diesel itself is a mutagen. I'm not sure of that. But I think it's a chemical dissolved in the diesel, which is the mutagen. And I suppose that's true for the cigarette smoke. There's chemicals in the cigarette smoke, which are the mutagens. Anyways, a mutagen is an agent that increases the chance of a mutation. You can also have a spontaneous mutation, and these mutagens occur in the absence of a mutagen. A spontaneous mutation occurs whenever the DNA is replicated and then a mistake is made. If the mistake is made and that mistake is not caught and repaired, it will result in a spontaneous mutation. Spontaneous mutations occur all the time because, what is it, something like 1 in 10 to the 9th bases that get copied. One will be a mistake, and then that can be a spontaneous mutation. So whenever we're replicating DNA, we replicate large numbers of nucleotides, and if any of them is replicated incorrectly, it will be a spontaneous mutation. Like I said, it's very rare. That spontaneous mutation rate is something like 1 in 10 to the 9th bases that are replicated. The trouble is, in a cell, we have more than 10 to the 9th bases, at least a human cell. There are different types of mutations. And here we're actually seeing a mutation in somebody's eye right there. That's why it's a unique eye color. There are different types of mutations. There are base substitutions. These are also called point mutation, where one base is mutated or changed. It can be 3 different types of base substitution. We'll talk about that in a little bit. There can be a silent mutation, a missense mutation, or a nonsense mutation, where one base is changed, resulting in the mutation. There can also be a frame shift mutation, and this is where a base is either added or subtracted, and you get the change in that addition or subtraction, but then that'll also change the reading frame for the codons. So if you change the nucleotide by adding or removing a nucleotide, all other codons after that point will be changed. And that's what a frame shift mutation is. And then there's a macro mutation. This is a topic which is not discussed by your book, so you have to get it only from the lecture. A macro mutation is where an entire region of a chromosome is either added or deleted. So it's a large chunk of DNA which is either added or deleted, and that's called a macromutation. If you look up macromutation online, there is another definition of a macromutation. There's the definition I gave, which you can find online, but there is also changing a regulatory gene, and they call that a macromutation because you mutate the regulatory gene and then it regulates other genes, and so it changes the gene expression of more than one gene, changes the gene expression of many genes. And don't get that confused with my definition of a macromutation, where you're changing an entire region of a chromosome being added or deleted. Does anyone know of a macromutation where an entire chromosome is extra?
Is it the Down syndrome?
Yes. And is that chromosome number 23? or number 21? I always get that confused.
I think it's 23. I don't know.
I think it's 21 actually.
I
Probably.
I used to know this, and to tell you the truth, I don't remember. Let me look it up. I'm just going to go online to look it up because I used to remember and I just don't remember anymore. It's not terribly important, but it's extra copy of chromosome 21. I should put that in my slide because I used to remember and now I don't remember. Where are we? Here we go. All right. This was a mutant or a possible mutant that I followed. This boy, if you notice, has spoons that were sticking to his skin. And they were testing him to see if he was a mutant that was causing this to happen. And there were two theories, and that is he was a magneto, where metallic objects were attracted to him, or the other theory is that his skin was sticky, and that's why the spoons were sticking to him. And like I said, they were actually testing this child to see if he was a mutant, but I never found the result, just that the study was in progress, and I never found what was the result of that study. So that's actually a possible mutant right there. All right, let's talk about base substitutions. Whenever the DNA is replicated, a mistake can be made where the incorrect nucleotide is added. In this case, instead of being a C, the T gets added. And now if this cell were to replicate, that T will get fixed because this DNA, the DNA polymerase will read that T and then insert an A. And when that happens, this mutation will get fixed in the population or in the cell. With this DNA being replicated, it'll be correct. So that will be replicated correct. And the cell could repair this if it notices that this is a mistake, because this A or this T will not pair with that G, and so that will cause a bubble right here in the DNA. And the cell can notice that, and it can fix this mistake. If it does fix this mistake, it always removes the newly replicated strand, meaning the T, and it just assumes that the T is the mistake, because most of the time it's the newly replicated strand which will have the error, not the old strand. And like I said, it can catch this mistake and then repair it. It'll excise that and then put in the correct DNA nucleotide. But if this cell replicates before that mistake gets fixed, that T will be fixed in the population, resulting in a mutation. Here, with this T being here, it should be a GC pair, and the GC pair codes for UAU, which codes for the amino acid cysteine. If it gets changed, it won't code for UGA, it codes for UAU, which will change the amino acid to tyrosine. And then this protein will be a mutant protein. Any question about any of that? Meaning the protein will get changed and it'll be a mutant protein. All right. One possibility is that when the base change happens, it can change the amino acid, which can be a silent or neutral mutation. So it's a base substitution, but if the amino acid gets changed and the change is from UUU to UUC, you change the DNA, but you do not change the protein because UUC codes for the same amino acid as UUU. And this is called a silent or a neutral mutation. Let's call it a silent mutation because the DNA is being changed. It's not changing the protein though. So a change in one base does not result in the amino acid change in the protein, so there's no effect on protein function. And that is a silent mutation. Any question about any of that? We can also have a missense mutation, which we've already talked about a little bit. And that is where you change the base, the nucleotide in the DNA, which used to be AAA, coding for UUU, which codes for phenylalanine. And I guess I read that wrong. I was looking where the arrow is. It should be CCG, which codes for GGC, which codes for the amino acid glycine. And then we change the DNA. Instead of having CCG, we have TCG. That changes the DNA and it changes the codon so that it changes the amino acid. Instead of coding for glycine, we now code for serine, which is the codon AGC. This changes the DNA, which changes the amino acid, resulting in a change in the protein, and this is called a missense mutation, where the nucleotide is changed and the amino acid in the protein is changed. One amino acid in the protein is changed. An example of a missense mutation is sickle cell anemia, where we change one amino acid in a protein containing thousands of amino acids, and it changes the protein so that that protein is more likely to sickle, causing sickle cell anemia. And we call it sickle cell anemia because the blood can sickle, meaning instead of being a nice red blood cell, it can sickle into an unusual shape. I suppose I should try and show you that.
Can I ask real quick? So the amino acid is added or changed?
In the missense mutation, you simply change the amino acid. It's not
Okay.
added. If you add it, it would be a frame shift mutation.
Okay, perfect. Thank you.
There we go. Here is a sickle cell blood cell, and there are some sickle cells here. They're a different shape, they sickle, compared to a normal blood cell shown here or here. All right, any questions about any of that? If not, it's time for me to end the lesson. So I'm going to stop here and I will see you at the lab at 6:31 or in the next lab at 2:30. Any questions? All right, I'll say goodbye and end this.